Hey there, science fans! Today, we're diving deep into the fascinating world of RNA Seq, a powerful technology that's revolutionizing our understanding of genetics. Imagine being able to take a snapshot of all the genes active in a cell at a specific moment. That's precisely what RNA Seq allows us to do. RNA, or ribonucleic acid, is like the messenger molecule of the cell, carrying genetic information from DNA to the ribosomes, where proteins are made. RNA Seq, short for RNA sequencing, helps us analyze the abundance and types of RNA molecules present in a biological sample, providing invaluable insights into gene expression patterns. It's like listening to the symphony of genes playing inside our cells. This cutting-edge technology has become an essential tool in various fields, from basic biological research to disease diagnosis and drug discovery. By studying gene expression patterns, scientists can unravel the complexities of various biological processes, identify disease biomarkers, and develop targeted therapies. It's like having a powerful microscope that lets us zoom in on the molecular machinery of life. Now, while RNA Seq generates a treasure trove of data, it's not as simple as just reading the results off a screen. Raw RNA Seq data can be like a jumbled jigsaw puzzle with pieces of varying sizes and shapes. That's where normalization comes in, acting as the puzzle master that helps us make sense of it all. Normalization is a crucial step in RNA Seq analysis because it helps correct for technical variations that can skew the data. These variations can arise from differences in sequencing, depth, library preparation methods, and other experimental factors. Imagine comparing the populations of two cities without considering their sizes. You might mistakenly conclude that a city with a higher population density is actually more populous. Normalization levels the playing field by adjusting for these technical biases, allowing us to make accurate comparisons between different samples and experiments. It's like calibrating a microscope to ensure that the images we see are sharp and in focus. Without proper normalization, our RNA Seq data could lead to misleading conclusions, like a blurry photograph that obscures important details. There are several normalization methods available, each with its own strengths and limitations. Let's explore some of the most common ones. Total count normalization, reads per kilobase million, fragments per kilobase million, and transcripts per million. TCN is like dividing the population of each city by its total area to get the population density. It normalizes the data by dividing the read counts for each gene by the total number of reads in the sample. While simple, TCN doesn't account for differences in gene length, which can affect read counts. RPKM and FPKM address this limitation by considering both gene length and sequencing depth. They normalize read counts based on both the total number of reads and the length of the gene. However, they assume that all genes are equally expressed, which is not always the case. TPM, on the other hand, is like comparing the proportion of people living in different neighborhoods within a city. It normalizes read counts to the total number of transcripts in the sample, providing a more accurate measure of gene expression levels, especially when comparing samples with different RNA compositions. In addition to these standard normalization methods, there's another technique called batch normalization that deserves attention. It's particularly useful when dealing with large-scale RNA Seq studies involving multiple batches of samples processed at different times or in different laboratories. Imagine trying to compare the results of a baking competition where different ovens were used, each with its own quirks and temperature variations. Batch effects are like those oven variations. They can introduce unwanted variability into the data, making it difficult to distinguish true biological differences from technical artifacts. Batch normalization helps mitigate this problem by adjusting for batch-specific effects, ensuring that the data from different batches are comparable. It's like recalibrating the ovens to ensure a level playing field for all the bakers. By removing these confounding factors, batch normalization improves the accuracy and reliability of RNA Seq analysis, especially in large-scale studies.